about to start having 2 talks. If Carrie will...
Shut up.
Yes.
Marta will arrive at some point.
Not even for your talk, and this is sad.
I wonder, hopefully someone will tell her where to go.
She has my mobile, but I'll switch the sound.
She'll be fine.
At the entrance, they will guide her.
Yeah.
And I'm very happy to have here from across the pond our speaker of today's 2 talks before lunch, and then we have a speaker from Oxford after lunch. We're going to hear about networks in the brain by Cardona.
Right. Welcome to the talk. It's so small that I think you should all ask questions right away and just get things clear as I go, rather than wait till the end. Also, the talk is more or less a half-hour talk rather than an hour talk, so there's plenty of time for discussion. So, I'm Cardona. I'm a group leader at Genelia and very interested in understanding how the brain works. So, to do that, basically to study the relationship between circuit structure and function, right? First, we need to know the actual circuit. You'd be shocked, but neuroscientists have been operating for a very long time without circuits. They just make up stuff in a way. They look at phenomena, they put inputs, the inputs correlate with the phenomena somehow, and they infer that there's going to be a particular structure that's doing a certain amount of processing, but they don't know the implementation details of that structure. And that's true for everything except the nematode, C. elegans, that was famously mapped here some 30 years ago. So, but the structure alone is not enough, even though Carrie can do a lot of things with it. It's just not quite enough. And so we need to observe as well the activity of the neurons over time to figure out the causal relationships between when one neuron is active or another one is inactive, and then look at the actual circuit activity. And that's not enough either. But we need to know as well how the circuit activity will change over time, which mostly has to do with neuromodulators that will change the excitability properties of the neurons and will completely shape the activity patterns in these circuits. And of course, it helps a lot if you have the ability to target the activation or the suppression of activity of specific neurons that you've identified and create neural behavior maps that Carrie and Marta so nicely published a couple of years ago. Because that gives you anchor points for either command neurons or neurons that are sufficient for a particular behavior or neurons that are necessary for a particular behavior. So that it's no longer a big labyrinth of neurons, but you have some lighthouses in there. And then with this data, we can formulate models on the basis of synaptic connectivity that can capture the circuit dynamics, and they can explain how the circuits produce the observed behaviors. And then we can test these models experimentally. That's the whole point. And so neuroscience as a field has advanced enormously by applying some or all of these techniques to very specific areas of the brain. But such as the olfactory bulb, the motor system, the visual system in mammals has been studied to death, right? But then you have to realize that no area of the brain works in isolation. There's fantastic papers from Michael Stryker, for instance, showing how even visual input is dependent on motor activity, that you can recover from visual loss, for instance, much better if you're actually moving. Somehow that has to do with lateral connection between systems, neuromodulation, and whatnot, right? And so all these areas of the brain are deeply interrelated. They don't operate in isolation. And so that's the key defining characteristic of the nervous system, right? That is, as a whole, the animal integrates all the information it receives from the world, visual, auditory, thermal, etc. And it has to integrate that with its own internal state, whether it's hungry, whether it wants to reproduce, etc. And then it has memories of past experiences. It has associated certain patterns of sensory stimuli with good things, bad things, with reward or punishment. Right? And then it has to coordinate its own body, because all the animal can do is move. And so motion in the end defines what the animal can do. And most animals have multiple segments, they're articulated, they have multiple joints. It's not trivial. And there's all kinds of interactions between different motor programs that are mostly suppressive. You can only do one thing at a time. You cannot move the hand both forward and backward at the same time, right? And So that already comes to show that there's going to be a lot of systems for decision making and action selection. And Marta will, I hope, talk about this much more than I will. But what I'm going to focus on here is on the circuit structures for learning and memory. And in particular, we've selected a model system in which we can study this at all these levels that I've talked about: the circuit connectivity, the activity of the neurons, the neuromodulation, and generate also neural behavior maps. And so this animal is the maggot, is basically the Drosophila, the famous fruit fly, not even the fly, just the larva of the fly. And in this larva, what we find is this tiny nervous system right here that contains about 12,000 neurons. It's tiny, but it's actually quite interesting in that every single cell is uniquely stereotyped and identified. If you go from animal to animal, we can find the same cell in the same segment expressing the same genes, and as we are showing in our papers, connected in the same way. This is very reminiscent of C. elegans, but at the same time, what we have here is a nerve cord that is structured in a way that is essentially homologous to the spinal cord of mammals. It uses the same molecules for guiding the axons in the mediolateral axis and the dorsoventral axis. I'm not going to name these molecules because I don't assume I have biologists in the audience. But even the genes that pattern the anterior-posterior axis of the animals are the same. So the homologies run very deep.
You said the neurons are completely stereotyped.
All of them except the ones I'll probably talk about today.
Did you say the
I did say it, yes.
Did you say the connections are stereotyped as well?
The connections, I have to qualify that. The strong connections are stereotyped. There's a low level of weak connections that is a lot more variable.
Now, is it true that you actually have never observed a second animal?
I've observed 4 different animals for various reasons.
Okay.
That's how I can make that statement.
Okay, thank you.
Big
Thanks.
data. Sorry, could you just define stereotype?
So by stereotype, I mean, say, what we have in here is one segment that has a complement of somatosensory neurons in your body wall, the skin, muscle, etc., and one complement of muscles. In this case, there are 30 on each side. So the exact same muscle has a name, that one muscle. And from animal to animal, you find that one again and again. And the motor neuron that talks to that is exactly the same motor neuron, expressing the same genes. And there's just one. You don't have a population that does that, you have just one, right? And then, so in the segments here in the nerve core, which are very compressed, and you don't see them, but it's essentially like the segments here, each of these hemisegments has about 300 neurons. And I'm saying about because I didn't count them, but each of these 300 neurons is exactly the same in the left and right side of the bodies and across multiple animals. That's what I mean by stereotype. It's what in the insect literature is referred to as identified neurons.
That's that one-to-one matching across. Now, did you once tell me that that won't be true for the adult?
The bigger the nervous system is, the more variation is going to be in number of cells. And that's because the neuroblasts are the neuroblasts are the stem cells that produce this temporal sequence of daughter cells. And there's going to be little slips in there where it might create an extra of this type of
More noise, the bigger it gets.
The bigger it is, yes.
You said they're the same on the left and right side of the brain as well?
Yeah.
Do they perform comparable functions?
Yes, identical. Yeah, which is the question then of how can you do asymmetric things if everything is symmetric, right? But that comes to function, and we'll get to that. Right, so this little monster, what's nice about it is that because it's Drosophila, we have an amazing genetic toolkit, right? And I will illustrate that later, what I mean by amazing genetic toolkit for manipulating the neural activity. But first, what I want to show is that it's small enough as well, that's only 100 microns, that we can image the entire animal with electron microscopy at sufficient resolution to resolve every single synapse between neurons. And what we have in here is a cross-section of the brain, and these little white, well, not white, but gray ovals, these are the somas, the nuclei of these neurons. And right here is the neuropil where all the synapses are happening. And so with this kind of volume, suddenly we can map from every muscle, every motor neuron, all the way back, and every single cell basically. So that was not done by chance, we were very fortunate to work with Rick Feta, an extraordinary electron microscopist at Janelia, who now has sadly retired, but he left the protocols behind, which is very good. And so he single-handedly generated a series of almost 5,000 serial sections of 50 nanometers each across the entire animal without losing any. Some were lost later during imaging. But to give you an idea, when I was showing a 500 set of sections to Mike Bate here in zoology in Cambridge, he was telling me that was impossible. So this was 10 times larger. He did it in 1 week, and for him it was not a big deal. He then generated a 10,000 section set for the adult fly brain. So that's coming too. That's what actually Alex's group, Greg Jeffers, is working on as well. And the volume itself is here in very low resolution. All of these are sections. Here we have the brain. Here's the pharynx going right through the brain. I mean, insects are a bit weird. And then as this comes back from anterior to posterior, this neuropil right here is where all the synapses are happening. This is the neuropil of the brainstem, and then it comes into spinal cord. And each of these little white lines that cross is some anatomical structure that defines each segment, basically. I won't go into details, but this volume exists, and it's structurable. And thanks to tools we had developed, we could take this pile of like 500,000 images and actually compose a continuous, homogeneous volume from which we could reconstruct circuits. And reconstruct we've done. This is a very collaborative project. It started in December 2012. And so over the last 4 years, we've mapped about 60% of it. It's very collaborative. My lab has done most in a way, but there are enormous contributions from labs in Germany, in Japan, in Barcelona, Marta's lab as well at Janelia, and many others. And so the areas of the brain that you see that don't, so each of these is a soma, that's a neuron. And the cable that you see here is visible because nobody has yet reconstructed that area in there. If this was fully reconstructed, all you would see is the somas of the neurons. So the same is true somewhere back here. There's still quite a lot of work to do.
Where's the mushroom body?
It's buried in there somewhere.
Okay.
All these small cells here are probably Kenyon cells almost. Yeah, the mushroom body is a brain structure, and genetically it has shown that it is homologous to the hippocampus, at least homologous to that same area of the brain. A beautiful work by Detlef Arendt, DMBL, if you care. Right, so the whole point of doing electron microscopy is that we can look at synapses. And so here we see just a three-dimensional cloud of all the presynaptic sites in red and postsynaptic in cyan. There are 1.1 million synapses at this point, and I expect at least another million to come. So it's going to be about 2 million, 2 to 3 million at most. And for anatomists, it is very clear which neuropils are which. I want to point out to you just to say it exists, we're working on it, and the areas that have holes is because we still haven't worked on it. So now, why is this an interesting model system to work on? It's because of the genetic tools. Not just the stereotypy, which is convenient, not just the size, which is convenient, right? It's really the genetic tools. And so Drosophila has made an art out of this. And by Drosophila, I mean the labs of Jerry Rubin at Janelia, Barry Dixon, and many others. And so this means that we can now label individual cells, individual uniquely identifiable cells, let's say a motor neuron here and a proprioceptive axon here. And then once we know how they look like from light microscopy, and we have maybe a behavioral phenotype for them when we activate them or inactivate them, then we can go to the electron microscopy. There's a cross-section of the nerve cord. And in that region of the neuropil, find that particular neuron and find that particular axon of the proprioceptive sensory neuron that senses the stretch of the muscles, and then reconstruct them. And so we end up with exactly left-right homologs, like I was saying, they're uniquely identified and mirrored across the midline. And then for those, because we have synapses, we can then reconstruct the first order network, all the neurons that connect to them. And so if you do that, you get with this enormous cloud of neurons presynaptic to the motor neuron, neurons postsynaptic to the sensory axon. And then very simple pathfinding algorithms can immediately give you polysynaptic pathways from the sensory neuron to the motor neuron. And Functionally, if you care about this is mostly about when the motor neuron never knows whether the muscle has finished contracting. It needs the stretch receptors to tell it, oh, I'm done now. And then this postsynaptic pathway is inhibitory, and it will suppress the motor neuron, right? That's something else to incorporate in models if you want, that there are many temporal scales that matter here. And now I'm getting sidetracked from what I was talking about, but I'll finish this. Say a motor neuron fires in 1 millisecond, but the muscle will contract in tens or even hundreds of milliseconds. So this temporal decoherence is re-coordinated through proprioception. And that's how the body then can adapt to going through water, going through molasses, going through ice, because you know how much effort you need to put to overcome whatever is on the medium, right? In any case, each of these cells is a uniquely identifiable cell. It has a unique name. We know exactly it's abdominal segment, lineage 31, K, right? It's exactly that cell. And then we can go to this amazing catalog of genetic driver lines specific for that cell that Jim Truman put together on Genelia, and then find a genetic driver line. So we have that particular line that will label all of the cells in the body. They're going to be left, right, in this case, in multiple segments of the nerve cord. And therefore, we are back to where we started. So now here we can do RNA-seq to discover all the messenger RNAs of that cell. Or, so we know the ion channels, the receptors, the visual transporters, the neurotransmitters, essentially. Or we can do neurobehavior maps. We can excite that cell, suppress that cell, figure out in what way it affects which behaviors, right? And so on. And of course, we can go back here and regrow the first order network for that cell. And that's the way in which we have reconstructed this 60% of the nervous system, not by going randomly, but by starting from cells for which we know what they do, such as sensory neurons or motor neurons, and then slowly growing the first order networks of those cells, and then second order, third order, and so on. And slowly by doing that, we'll cover the whole brain, but always with a handle on in a system that we can understand, basically. Right, so now let's move on to learning and memory, because what I want to talk about is circuits for learning and memory. And the first point to make is that maggots can really learn. And by learning, we mean associative memory. There's this very simple assay, thanks. Someday I'll learn to speak properly.
Say it in Spanish.
Ensayo. Right. So what this does is they look at the performance index. And this is computed by having two sets of experiments, one in which you pair an odor with a reward, and another one in which you present first the reward and then later the odor, or the other way around, depending on various. And then later you test in a simple plate. Half the plate will contain the odor, so to speak, the other one doesn't. And then you test whether the animals congregate on one side or on the other of the plate. It's a very simple assay. But essentially by combining these two, if they really didn't care, you would have a performance index of 0 if they had not learned anything. If they learned to like it, you would have a performance of 1. And if they dislike it, it would be -1. It would be all on the other side. And so when you do that for maggots, of the first instar, the smallest newborn maggots, they already show the ability to learn. And this is a box plot, and I apologize for that, it's not from my paper. But basically it's about 0.2, 0.3, they're always very low, but they are significant. So the effect is there, even if it's weak. And it can be done much stronger now, thanks to new tools that Andreas Thum has put together. But this is actually from Andreas Thum lab as well. So the ability to learn has been located in insects in the mushroom body. The mushroom body contains these cells called Kenyon cells, which are parallel fibers. And these cells are both necessary and sufficient for learning. If you inactivate these cells, you cannot learn. And if you activate them, you actually can induce memories in their various ways. And so here's a summary of what the mushroom body looks like and what it does, from what we know from the literature. So what I'm showing here are sensory neurons, in this case olfactory sensory neurons, that very specifically connect to projection neurons that integrate those inputs, and then the Kenyon cells, the dendrites of the Kenyon cells randomly sample from the projection neuron axons so that they create this, they increase the dimensionality of the representation by mapping this, say, 21 vector input into like 100 or so. This was shown with light microscopy in the adult. We have now shown this using electron microscopy. That is also the case. Because we don't find Kenyon cells that are the same between left and right, except for a small subset that I'll talk about in a moment. Then, so these Kenyon cells have these axons that run along across multiple compartments. This is very exceptional. Generally, axons are not compartmentalized in that way, but here they are. And who creates the compartment morphologically is a combination of both the axons of the reinforcement neurons and then the dendrites of the output neurons. Both are extremely sharply localized into a single compartment that is crossed by these axons. And these reinforcement neurons encode for various kinds of punishment and reward. And then therefore, what's expected to happen here is that the combinations of Kenyon cells that are active somehow are learned by changing the weight of the connections onto the output neurons. And this weight is changed if there is activity from the reinforcement neurons, which are dopaminergic. And then the output neurons by themselves, if you ignore the rest, if you activate any of these neurons, you can see that they encode valence and guide memory-based action selection. Like the activation of specific MBONs artificially using optogenetics leads to either promoting avoidance or promoting approach towards an odor. And so we find that the compartments then are specialized. Some are specialized for reward, for things that the animal learns to like, and things for the animal learns to dislike. And then how these work?
Based on which output neurons?
Yeah, based on which output neurons. So the output neurons by themselves already are stereotyped in what they encode. But now what changes is the weight of the connections from the Kenyon cells that randomly sample from the inputs onto these output neurons. And this is meant to be a synaptic depression. And so how this works, I'm not sure, so how this works very briefly is that these neurons do the opposite of what you would expect. So if you want to learn, if you learn to like something, what you're actually learning is to avoid it less. That's why you repress this reaction. That's how the system works.
That's what I kind of think of like as well when it comes to other humans. I mean, I don't like them so much, but I'm willing to avoid them less.
Right, and then if you learn to hate something, you actually learn to like it less. That's also how it works. So the dopaminergic neurons that encode reward are in the same compartment as the output neurons that, when excited, induce avoidance. And the opposite. If this encodes punishment, this encodes approach. And synaptic depression makes sense, right? That's all you need to know about the Martian body carry.
Okay, good. Why do they do it that way?
Probably because of this. So a recent paper in eLife has shown that each compartment has different memory characteristics. Some are for short-term memory, some are for long-term memory, and all of them have different extinction rates. So it is somehow, for some reason, easier to create synapses than to pull them out, I guess. And so by default, the system is trying to connect.
Okay.
And it is only, so you forget quickly. Whereas it's a reinforcement that makes you forget by actually pulling out, pushing down the weight of the synapses. That's how it works. Right, so now the question is, if these systems look so similar functionally, what kind of microcircuits do we have in here? Are they all different as well? Are they all the same? Do you have a generic microcircuit that will encode the memory formation, and therefore it's completely independent of whether these output neurons encode for approach or avoidance? And I'm saying that because that's obviously the case, and I'm going to show you in a second. So this required mapping this. It looks weird, it's a bit off, but mapping the wiring diagram from the electron microscope. What we see here in black are all the axons of Kenyon cells. It's kind of outside the image, a bit unfortunate. Here is the olfactory system in glomeruli, like ours in the olfactory bulb, that then projects up into this particular region called the calyx right there, where the dendrites of the Kenyon cells are randomly sampling from these inputs. And then the compartments will be along this, I can't really show it, so I just leave it.
But that right there,
Yeah.
forgetting the compartments, is the adjacency matrix.
Well, it's the connections between the cells, yes.
And what's the sort of thin
Oh.
film that looks like a spider web, right? Closest to the center?
So, yeah, fair enough. That is the outline of the entire central nervous system that I had put there as reference, but the contrast is a bit off somehow. It's barely visible, right? You see it here. It doesn't matter. This is anatomy. Let's not go there.
Okay.
What's interesting is this. Because we completed the reconstruction of this system, we know all the projection neurons for various systems. This is from olfaction, from gustation, some we don't know, some from encode temperature, others visual inputs, and some encode combinations of that. And then what we find is that there are Kenyon cells here that integrate inputs from either 2 of these, or 3 of these, or 4 of these, or 5 of these at random. Because when we look at left and right, we don't find the same combinations.
I missed what is a claw?
Right, so a claw is a dendrite of a Kenyon cell. Yeah, thanks. So these Kenyon cells are very small dendrites, and generally they are directed into grabbing input from one single axon button. And so they look like little claws because they wrap the axon button like this. And so this one here would have 4 of these grabbing 4 different axon buttons of the projection neurons that bring in the sensory information into the system. So the surprise was that we found in here a subset of single claw Kenyon cells. And so these are stereotyped. And they're stereotyped because in the left and right we find the same ones. So this was really, really surprising. It's not for all the PNs that we find that. It's mostly for the olfactory projection neurons. Why that is, I don't know. But that's what we see. And a summary of this would be the following. Now we're looking at left and right. So this is left, this is right for every single one. This is the number of dendrites, number of claws, which amounts to the number of different projection neurons this Kenyon cell is integrating. And so here we find in blue are the common ones. So there are about 13 that are common between the left and right. 12 actually. 12 of them. Others are not. But then if we look at all the others, they have more than 1 claw. Between the left and right, we never find the same combination. That's what was known before. The surprise was the addition of this few set. And what's interesting is that these few set are the earliest born cells in this system. And the earlier born cells are generally special in some way. That's a whole other discussion. We can have that. But now the question was, wait a second. So this animal has the expected system of random sampling from the inputs of the sensory systems, but also a stereotype system that just specifically picks from one, right? Symmetrically between left and right. Why would you want to do that? So we're fortunate to work with Ashok Kumar in Larry Abbott's lab, and he created a classification task for odor discrimination. And what we're seeing here is the error in the odor discrimination. And here, basically a number of simulations in which he changes the mean number of claws of these Kenyon cells, and how that would affect the ability of the system to discriminate different but similar odors. And so it turns out that the best possible configuration is actually the observed one. And so it turns out that this combination of random sampling and single claws is making this animal cleverer than it would be otherwise, at telling apart similar inputs. And I'm not showing it here, but he also demonstrated that this is very dependent on the number of Kenyon cells. In this tiny maggot, this early born maggot, there's only about 50 or 60 operational Kenyon cells on each side, maybe 70 if you stretch the connection, right? And whereas in the adult, we have 2,000. And so in his simulations, he shows that if you have 2000, you don't need the single claw ones in order to have better discrimination. But if you don't, having these label lines somehow is beneficial for the system to be able to tell apart things. The math is all in the supplementary figures of the paper here. Just showing you the graph. Right, so we have an explanation at least for why this system would be built the way it is. So now we move on about the structures of compartments. All of these compartments seem to be doing the same thing. They do associative learning using synaptic depression. What kind of circuit supports that? So what was known is that the sensory stimuli excites the Kenyon cells that excites the output neurons. These are the parallel fibers. And what was also known is that a different kind of sensory stimuli, like pleasant things or painful things, are able to drive the reinforcement neurons that somehow affect the Kenyon cells. That's what was known from a functional point of view. This is not a wiring diagram of connections. But now this is a wiring diagram of connections. And it turns out that in every single compartment of the machine body, we find exactly the same circuit, which is perhaps not surprising. They're all doing the same, and the valence of the outcome is really encoded by the structure of the output neurons, where they project to. But what we find here is a number of very interesting connections. So these connections were known, this connection was known. So the surprise here was that you also have a direct connection from the reinforcement neuron to the output neuron. Like, why in the world would you want to do that? And so already some preliminary evidence unpublished is showing that this is about the preparatory actions. So in learning, there's a preparatory action and a consummation action, right? If you learn to associate an odor with reward, when you sense the odor, you prepare yourself for consuming the reward. In this case, preparing yourself means you're gonna, for instance, chemotax until you find the reward, right? The food. And so when you find the food, you need to stop. If you don't stop, right, you can't consume your reward. So who does the stopping? Well, it is known that these neurons will fire when you find the reward. And it's known that these neurons do the opposite of the preparatory, right? You're not avoiding, this is where you had learned not to avoid it, right? So by exciting this, you will avoid it somehow, right? That might make sense at some point. It's just too much because I'm not showing any functional data for it. I'm just telling you that.
No, I understand the story. Yeah.
People are working out a hypothesis on why that is, and now they'll test it.
But it's there, and
it's there
had
consistently.
never been seen before.
Had never been seen before, and it's consistent in every compartment. And suddenly now it makes sense if you think of the consummation of the reward and the preparatory actions towards that consummation. And in discussions with people in the field, I told them, wait, and wouldn't that be as well an explanation for, say, obsessive compulsive disorders, where you go and open a door 10 times? Like, you're doing preparation, you're not actually grabbing what's in the drawer. It will be some disorder that affects this kind of connection, right? Because all of this is very similar when you go to cortical structures in mammals, or the dopaminergic system interacting with the output system and their parallel fibers and whatnot. Then the other interesting bit is the parallel fibers talk onto the axons of the reward neurons. Again, why would you want to do that? Well, functional data shows that after learning, the reward neurons stop firing to the reward at the same intensity. Because what this really encodes is the expectation of a certain amount of reward. And so these neurons will stop firing to the reward when it's at the same level as you had seen it before. But they will start firing now to the particular percept that was associated with that reward. So one possible way where that could work is if this synapse was plastic and you had learned now that when you perceive these, you make these neurons fire. Right? How that relates to the other story, I don't know. But that's one possible direction in which you can maybe make these neurons fire when you have learned something. Because this is known. These neurons will stop firing to the reward, but they will start firing instead to the percept associated with the reward. And then finally, notice as well that these connections are onto both the presynaptic side and the postsynaptic side of this connection here. because that's axon to axon. So these three synapses, 1, 2, and 3, are all within the compartment and spatially within a micron of each other. And if you can affect both the pre and the postsynaptic side, suddenly you're posing a situation where spike time independent plasticity, like three-factor Hebbian learning, could be operating here. I'm not sure how that plays along with synaptic depression, but that needs to be worked out. But it's an interesting possibility that you're controlling both the pre and postsynaptic sides at the same time.
But the stochastic process on this network is quite a bit more complicated than, or the network upon which the stochastic process lives is quite a bit more complicated than previously thought.
What do you mean by the stochastic
So,
process?
what you have here on, so now if you go back to the actual
Yes.
network,
Yes.
you've got a process giving the firings and whatnot.
Yeah.
And-
Well, the network is much bigger than this.
Right, I know, but the idea is that if you erase the edges that weren't previously known, then the ability to understand the process is going to be quite,
Yeah.
oh, okay, this is not going to be so hard. And now when you put your new edges in there, the process is just going to be a mess. So you should take them back out.
That's exactly what Barbara Webb said.
Yeah.
It was a modeler who
Yes,
was
that's
dealing
right,
with this
that's
and said, oh my God.
This makes the math harder, so let's pretend it's not there.
Yeah, but that, unfortunately, we can't do. So this is just to show every single compartment has the same combination. And even when you have multiple reward neurons, you still have the same, always the same setup of connections between the reward neurons, the parallel fibers, and the output neurons. It's quite something. It really is the mechanism by which learning is somehow operating. Now it needs to work out how it works. So, but of course, the network is not limited to what happens within the compartment. There are plenty of interactions across compartments. And again, ask yourself, why would you want to do that? It's because it would make no sense to learn that you like something and dislike something at the same time. You have to have, you either like something or you don't. In the simplified world of the larva, where you either approach or avoid an odor, okay? And so what we found is that, so what I'm picturing here, each of these nodes is a neuron, so these are connections between neurons. All the neurons that have a G are GABAergic, so they're inhibitory. And what we see is a lot of connections between, say, these 4 compartments here, which are known to mediate reward associations. Like you learn to dislike things less here, okay, and you learn to goodness, the double negatives are crazy. And here you learn to avoid things. No, actually, you learn to approach things less. Exactly. Yeah. So between these two systems, there's a lot of negative interactions between them. That makes sense, and it makes sense even further because remember, suppose you learn to dislike something less, like here. You create an association here, right? So this will have the effect of first this cell, whoever is talking to, it will be less active, right? So it will be inhibiting less whatever is downstream. But at the same time, it will inhibit, say, this cell. And this cell is the ones that are, when excited, encodes approach, which is in the other system. So it will disinhibit approach. It will by itself be less active, and by being less active, one of the effects is that the cells that encode for approach will be able to be more active. And remember that the parallel fibers talk to all of them. And therefore, this cell will get probably more excited when you have learned an association here, because these cells are less excited, right? So the interactions are quite complex, and they're not fully symmetrical, but they are stereotyped between the left and right side of the brain. The same identified neurons, here I'm just showing you one side of the brain, are connected in the same way. And so this matters to the larva somehow, and we haven't yet figured out all the details on how. But it's obvious already that the interaction across compartments at the level of the output neurons are very disinhibition, by enabling things to happen. This is merely the connectivity between the output neurons of the compartments. Now we can look at feedbacks and feed across. So for instance, there's going to be output neurons that also talk to the reward neurons. Remember before what I said is the reward neurons talk to the output neurons, right? But in some compartments you have the output neurons also talking. The axon of the output neuron talks to the dendrite of the reward neuron. I'm not going to speculate why you would want to do that, but just to show you that there are various patterns, and this is all that we found at the first order. And so some are within the same compartment, some are across compartments, and some are even across compartments of different systems, like the say for reward and for punishment. So here's a compartment where you learn reward associations, and it turns out the output neurons of some of the compartments for punishment associations are interested in talking to that dopaminergic neuron. Why would you want to do that? I don't know. We'll have to figure that out, right? But this is all related to this ability to not have conflicting memories, most likely. That would make sense. It's the obvious thing that comes to mind. So that's only the first order connections between output neurons and input neurons. The real meat of the matter is in the second order connections. There's a gazillion amount of cells that are postsynaptic to the output of every compartment, and then they are presynaptic to the input, to the reward neurons of every compartment. And on top of it, many of these feedback cells, as we call them, synapse to each other. And many of them are inhibitory. So you're going to have a lot of inhibition and disinhibition going on that enables or disables modules of this system. Working this out is something Marta is very interested in and focused on at the moment. I don't know to what extent, if she comes, we'll talk all about this. So I've talked about this. I think what I want to show you now in the last few minutes is a cautionary tale in the interpretation of these connections by bringing in some anatomy into here. This is a conversation we started before. So as I said, many of these connections within every compartment happen spatially very close, sometimes even that close. And so you have here, say, the reward neuron that is synapting directly onto the Kenyon cell and onto the output neuron, and then these two connect to each other as well. This one is very close, some are just within half a micron most. And what does that mean, right? If we look at that carefully, what does that mean? Well, it means that this is probably a very misleading statement. When you look at two neurons that are parallel fibers, that they synapse onto each other, and you say, well, the tissue matrix shows there are 11 connections this way, 3 connections that way, and you go and do your inference, and you're done, right? But these connections here are not of the same kind, and that is something you have to consider. So for this, I'm going to go out and do something terrible, which is do a demo. And what we see here is these two cells, one in orange and one in blue. And so in the first place, these are the dendrites here. There's always, these are the somas, the dendrites. This is where they integrate the sensory inputs via projection neurons. Then here's a linker segment, it's the action initial segment where the spike initiation happens. And then the spike will run all the way through these lobes. And so if I rotate this in space, this is the medial lobe, that's the vertical lobe. So this one is all for learning punishment associations, this is all for learning reward associations. And this compartments here, we have no idea, but eventually we'll figure out. And so the spike will run this way, but it will also run that way, a backpropagating spike. And in here you have dendrodendritic connections. And so the compartment here will communicate, Kenyon cells will communicate to each other in two ways. Via graded potentials, as the integration happens in the dendrites. And then the backpropagating spike will come back and also elicit this charge. That's Marta. Right, whereas here, what you have is axoxonic interactions, very localized within the axon, right? So let's go back here. here's where we were. And now what I'm gonna do is explode this neuron at this single node in the graph by adding anatomy to it. And so here we would have the dendrites, the dendron-dendritic interactions between one cell and the other. And then at every compartment, you will have none or two or one connections, very weak, right? So what does that mean? It means first that these connections are dendron-dendritic and they're qualitatively different to start in the two ways that I mentioned already. And then these connections here, if they're very weak, they're gonna be, so weak connections provide weak depolarizations potentially. This needs to be measured, and weak depolarizations are subthreshold. They don't make the other neuron fire. All they do is they locally diminish the voltage potential. That has the effect that if now there is a spike coming, you will discharge less at that synapse. So in a way, this excitatory weak connection is a form of inhibition. At the same time, if the connection is sufficiently strong, like maybe these three connections here, the depolarization might be above threshold. So maybe you have now an excitatory interaction right there in this particular compartment. So the same synapses between two neurons maybe doing three different things. Maybe these weak depolarizations are inhibitory. These strongish connections might be excitatory. And the dendron-dendritic ones will maybe fire twice to gradient potentials and to vapor-propagating spikes, right? What that means, I don't know. But it's the cautionary tale that the adjacency matrix can be very misleading when it comes to particularly Kenyon cells. In general, separating dendrites from axons might be sufficient to understand most of what you need to know. But in Kenyon cells, because the axon is tiled in this very peculiar way, and the connections within a compartment matter a lot for learning, you have to consider that there are these complications. So a Kenyon cell is really a multi-node neuron. It's not just one block. Okay?
So beyond just whether the edges, if you colored the edges axonic and dendritic, that's one thing.
That's one thing, which works for most neurons.
But for Kenyon cells, you need to go beyond that, and you need to color edges by compartment?
By compartment and strength.
Compartment and strength, okay.
Because the strength might have inverse effects, big versus small.
Okay, so we can do that.
I'm sure you can. It's a matter of can we make sense of the result.
That's your problem. But compartment and strength for the Kenyon cells, the Kenyon to Kenyon cells.
The Kenyon to Kenyon cells have this additional complication.
Yep. Okay.
Yeah. And the same is true about the Kenyon cell to DAN connections. Okay. Weak ones versus strong ones may have very different effects. Yeah. And you see the whole distribution of weak to strong.
Right, and I saw that a difference if you sort of ignore weights versus don't ignore weights.
Sure.
A localized difference, so that might be what you're saying. And I saw a difference if you do, color by
axon and
dendritic, but I haven't looked at the compartment yet. Because I only learned about these compartments in reading my paper.
So the key point of the compartments is that each one is either for reward associations or punishment associations, and different kinds of rewards, different kinds of punishments, and with different properties, like different extinction rates, short-term memory, long-term memory. So really they are different. And these differences matter a lot to the actual.
So from a math perspective, a math on graphs, statistics on graphs, this cautionary tale is just, it's not so much against the adjacency matrix, it's saying you have to color the edges and then figure out how to do your inference on more complicated.
It's basically saying
Marta, what you called your multicolored edges. I mean, Sophia.
But it's basically a weight, isn't it?
But he's saying there's weights and there's also these compartment things, which would be like colors.
But even
categorical, as far as I can tell. He means
I mean the spatial location matters. Along the axon. And then the weight within matters as well, and that it may have opposite effects.
Right. So it'd be a multi-layered or a multi-graph with weights as well.
Or you could think of it as
And
just
the directions are absolutely essential.
Or you could just have an edge
Oh, you already had
covariance.
directions.
Yeah, I already had directions.
Or you could just have edge covariance.
Right, that's what I'm saying.
I think here it would be easier to have edge covariance rather than think of it as a multi.
So that's a good way to put it, is that if you're just adding a little bit, because it's two ways to think of the same thing. If you're just adding a little bit, but he seems to be trying to pile on complication after complication, pretty quickly you want to just go to the covariate version. Yeah.
It's not complication, it's the fact that if you meddle these things together, the system makes no sense.
Right.
But it's also like multi-compartment models of neurons.
Mhm.
I mean, that's really what you're talking about. There's different compartments. A single neuron is ultimately 3 or 4 neurons.
Right.
And it does different
Well, in this
processing
case,
in different parts of the Abbott.
There
That's
are
all it
11
is. It's
compartments
quite
in the
simple.
natural body.
Right.
So there are 11 divisions. One division is this one where the dendrites talk to each other. The other 10 are all equal in that they're all axonsonic connections.
Right.
But then locally, the strength will actually have a huge effect.
Yeah. Right. Very good.
Okay, so I have maybe one more slide and that's it.
That's fine.
Is that good?
Your time, I think, on this. Yes. perfectly on time.
Okay, then the one more slide I want to show you is kind of already a big view of how the brain works when it comes to connectivity. So we always have something that reminds me of a U-net architecture. Are you familiar with a U-net? It's this artificial neural network that first has a lower resolution and it divides by 2 and it's, goodness, look it up. At every level of the layers, of the deep layers, say the outer layer talks to the most outer layer on the output side, and then the second author also talks to the second author, and the third author also talks to the third author, and each one in the middle, it has less and less resolution in a way. That's why it makes a U like this with all these connections. I should have put it down again. So you find that because you find that the sensories talk to the projection neurons that talk to the parallel fibers that talk to MBONs that then descend back. So this is the deepest part of the U, so to speak. But then each of these is also, in various ways that I'm not even showing here for this particular system, immediately talking as well to the intermediate parts and to the output parts.
So what's the significance of the U?
So the significance of the U is that it works amazingly well as a neural network. It outperforms many other neural networks.
So it's just loops, having multiple loops.
Yeah, you have loops.
Short, longer.
The
That's
sensories
all
talk
it
to the
is.
motors, and when the sensories talk to the first order also talks to the motors, and when the first order talks to the second order, the first orders are talking to each other, the second order are talking to each other, they all talk back. So you have this ladder-like in which you descend and you ascend, but at every step of the ladder you're also talking. And that's what you find throughout, which is interesting. I was,
So if you had to reorganize this picture as a new,
yes.
how would that go?
Yeah, what happens is there are many entry points, and that's the difficult part. Like from, say, mechanoreception talks directly to motor neurons. The proprioceptors, for instance, talk to motor neurons. This talks to the premotor neurons, they also talk to motor neurons. But then these premotor neurons, many of them actually are of this kind. They talk to higher order brain structures that also talk back to them. And so you have this U here, and this U here, and then this U here, and the even longer U's via the mushroom body.
So it's a U with more than 2 stems, I guess.
Well, there are many
Yeah.
overlapping U's,
Okay.
depending on, but they all have the same output stem, which is the motor system. Whereas the input stem is, there are many.
There are lots of
Faction,
them.
vision, tonic sensation, etc.
Oh, that makes sense.
Okay, good. I'm glad it makes sense. I'm done. Just to acknowledge that this is the work mostly of Kathy Eichler and Feng Li and Asha Kumar, which is not here, oops. And in collaboration with Gary, with Larry Abbott, and Marta, of course. Marta did a lot here too. So thanks for your attention.
So we've done questions to the talk, but are there more?